The overall goal of this procedure is to generate whole genome DNA replication timing maps from zebrafish embryos in different stages of development.
This method can help answer key questions in the cell cycle and chromatin biology fields, such as how chromatin changes that occur during embryonic development can affect the initiation of DNA replication forks. The main advantage of this technique is that it maps replication changes that occur in vivo at defined stages of embryonic development.
Though this method can provide insight into how DNA replication changes during zebrafish development, it also can be applied to other systems or model organisms such as tunicates, frogs, and flies.
Generally, people new to this method may struggle because it does take some practice to generate single-cell suspensions from zebrafish embryos.
The night before embryos will be collected, place dozens of breeding adults into breeding tanks using approximately equal numbers of males and females. Depending on the experiment, use one of the following breeding strategies. For the first breeding strategy, place a few male and female zebrafish in individual breeding tanks and use a divider to separate the males from the females. If this approach is used, set up many different breeding tanks and pool the embryos from each to ensure the biological variability is representative of the population. For breeding strategy number 2, combine dozens of male and female zebrafish in a large breeding tank. Use this strategy as long as there are a sufficiently large number of embryos. It is reasonable to assume they came from a variety of founders and are therefore genetically representative of the population.